Okay, so what we're going to focus today is about enzymes. What enzymes are and what they do, structure of enzymes and how they work, as well as the factors affecting enzymes activity. Okay, so enzymes speed up biological reactions, specifically chemical reactions without being used in the reaction itself. So this means they act as a biological catalysts. So enzymes are protein that speed up the reactions or catalyzes reaction. So enzymes don't make reactions happen though. Always remember that. They can only speed up reactions that already occur because they didn't get used up, they can be reused. A small amount of enzymes goes a long way. Okay, so starch is used to store energy, but in order to make use the energy, we need to break it up. Okay, so here's the enzyme amylase. Amylase is produced by the salivary glands in the mouth. So it speeds up the breakdown of starch. Since it works within the saliva or outside the cell, it is called extracellular. Okay, so like the enzymes involved in collagen synthesis known as hydroxylases. So in any case, hydroxylases catalyze the build-up of collagen, specifically the addition of groups, which allows the helical structure, this one, on your screen, to form. Okay, so enzymes are involved in a huge range of reactions. We have metabolic, for structural, breakdown, synthesis, and inside and outside of the cell. So they play indispensable role, and the key on how they work is their structure. Okay, so enzymes are type of globular protein, several polypeptide chains bound together to form a shape, this one, or the enzyme. So somewhere within the shape is an active site. Okay, so we can call it interactive site where the enzyme interacts with other molecule. So like all protein structure, the 3D shape is determined by the tertiary structure, and it's so important to get this right because it is highly specific. Okay. Any substances that interacts to the enzyme do so by recognizing and binding to the active site. So they are known specifically as substrate molecule. This one. Okay, so if they are bind together to the active site, they will create enzyme substrate complex. So always remember that enzymes will only bind to substrate that are perfect match. Okay, so there are things we know and the things we don't know about how enzymes actually work. Okay, so enzymes speed up reaction by reducing the amount of energy required to start the reaction. It is known as the activation energy. Okay, so imagine the activation energy as the hill or a barrier. So kailangan mong mag-exert ng effort para makarating ka sa peak ng hill, just like what enzymes do. So in every reaction to get from substrate to product, the activation energy must be reached. So enzymes lower the activation energy slightly so that less energy is required for the reaction to start. Okay, so enzymes lower the activation energy slightly by either bringing the substrate together in a joining reaction, or they can strain the substrate bonds in a breakdown reaction. Okay. So energy is often provided in the form of heat. So specifically, enzymes allow reaction to take place at lower temperature than usual, such as our body temperature. So without them, many important reaction would proceed too slowly to sustain life. Okay. So enzymes don't trigger reaction. They make it easier for them to happen by lowering the activation energy. Okay. So how do the active site and substrate come together to lower the activation energy? So scientists have these two theories, the lock and key model and induced fit model. Okay. All right. So, as the name suggests in this theory, the enzyme is like a lock and the substrate is like a key. So, the substrate lock in the enzyme active site like a key fits into a padlock. And this formed an enzyme substrate complex. So the reaction produces an enzyme product complex. So imagine, tinwist nyo yung key sa padlock. What will happen to the key? So it will be broken. That broken pieces of key is the products. It is the products, and they are released from the enzyme. It remains unchanged and the process happens all over again. So this theory, the enzymes and substrate are perfect match. So the only problem in this model is that the enzyme doesn't seem to have as the shape as they suggest. It actually even shows that the enzyme substrate complex change its shape slightly to complete the fit of the active site. Okay. So we have here the meaning of the two theories, the lock and key model and the induced fit model. Okay, so let's proceed in this slide. Okay, so in line with this event, balik tayo sa structure na to. So in line with this evident, the included fit model is developed. So in this model, the substrate locked in the active site of the enzymes before, but this time the enzyme substrate complex is slightly changed its shape to make the fit even better. So it's kind of like a pair of new shoes. So you need to stretch them out a little so they fit just right. So it's a similar thing with enzymes. Basically, in this fit model. So the active site molds itself around the substrate. It's still a specific pairing, but the active site changes shape to fit even more. And as before, the enzyme's product complex is produced and the product is released. Okay. All right. So enzymes are really important. We can't live without them. Let's move on with the factors that affect enzyme activity. I have here a picture of a sliced apple. If you allow to sit untouched, the flesh of sliced apples will turn brown by a process known as oxidation. And it is actually caused by an enzymes. So when an apple is cut open, an enzyme or the protein that causes chemical reactions called what you call this, the polyphenol oxidase. It is released from the cells of the apple and reacts with the oxygen in the air. So this reaction causes the fruit to turn brown, similar to rust forming on metal. So almost all plants contain polyphenol oxidase. So it's believed plants use it for defense. When plant is damaged, the browning of the affected area was discouraged animals and insects from eating. It may also work to heal the plants. So the browning creates an antibacterial effect, limiting the spread of destructive germs. Okay, so here, so lemon juice, however, uh, acts as a preservative. Why? Because it's full of ascorbic acid. So ascorbic acid, we all know that it is an isolate of vitamin C. And it has a low acidic or the pH level. So ascorbic acid reacts with oxygen before it reacts with polyphenol oxidase. So even when the ascorbic acid is used, the pH of the lemon juice prevents browning. So that's because polyphenol oxidase works best in the 5 to 7 range on the pH scale. And so lemon juice's pH level is about 2. So which keeps the polyphenol oxidase inactive. If lemon juice is sprinkled on the sliced apple, the vitamin C in the lemon juice will inhibit the formation of this brown color by changing the pH of the environment of the enzyme. Okay, so enzymes reactions are affected by reaction conditions like pH. So pH is the level or the percent of acidity in an object or food. Okay, so enzymes are most active at a pH known as the optimum pH. So at optimum pH, the enzyme maintains its tertiary structure and its active site. Okay. And so the optimum pH for enzymes is based on the location of the enzymes as shown in your screen. So we have here the pepsin, which is located in your stomach, and the substrate is the peptide bonds with the optimum pH of 2. So we have here the sucrase, the urease, the hexokinase, the trypsin, and the arginase. Okay. So other factor that affect enzymes activity is the temperature. So like in pH, enzymes have an optimum temperature at which they are most active. Okay, so the optimum temperature for most human enzymes is normal body temperature, which is 37 degrees Celsius. Okay, so what will happen if there is an increase and decrease of temperature? So at high temperature, enzymes denature, which modifies the active site. Ayan. So while at low temperatures, enzymes activity is low due to a lack of energy for the reaction to occur. So just like for example, food is stored in a refrigerator or freezer to slow spoilage brought on by enzymes. So one of the example again is the boiling. So boiling contaminated water will destroy enzymes in bacteria that are present in the water. So there is a picture of the natured um, um, there is a picture of the natured enzyme. As you observe, there is a change in shape. So the enzyme lost its original form, and when the enzyme lose its shape, automatically the enzymes will lose their function. Okay, so last factor is inhibitors. So there are types of molecules that will cause enzymes to lose their activity. So enzymes inhibitors prevent the active site from interacting with substrate to form ES. Okay, or the enzyme substrate complex. So some inhibitors cause temporary loss of activity while others cause permanent loss of activity. Okay. So when we say reversible inhibition, it occurs when the inhibitor causes a temporary loss of activity. However, activity is regained if the inhibitor is removed. So it can be competitive or non-competitive. All right. So as long as an inhibitor remains in the active site, the enzyme cannot react with the substrate to form a product. Okay. So an example of a medical therapy that involves a competitive inhibitor involves the liver alcohol dehydrogenase, or what you call the LAD. So this enzyme oxidizes ethanol, the alcohol found in alcoholic beverages. This enzyme will also react with the ethylene glycol and the methanol, which are found in the antifreeze. So the antifreeze is a liquid that yung nilalagay sa mga makina ng car para ma-maintain yung, or para hindi masyadong uminit yung makina ng isang sasakyan. Ayun. And all right, so this enzyme will also react with ethylene glycol, and the methanol, which are found in the antifreeze, and will compete with ethanol for the active site. So if a pet is poisoned by drinking this antifreeze, a slow intravenous infusion of ethanol is administered. So, administration of ethanol slows the production of the toxic metabolites of ethylene glycol and methanol, giving the kidney time to eliminate these two substrates. Okay, so these figure diagrams show how a non-competitive inhibitor functions. So non-competitive inhibitors do not resemble the substrate. They do not compete for the enzyme's active site. So non-competitive inhibitors bind at a site on the enzyme that bind on the site of the enzymes that what you call this that is actually remote to the active site. So when a non-competitive inhibitor binds to an enzyme, it causes a conformational change in the enzyme. So this change in shape causes the active site to no longer interact with the substrate. Okay. So as long as this type of inhibitor is bound to the enzyme, it will no longer function effectively. So as what you can see in this illustration, here is the substrate and this is the enzyme. This is the inhibitor, specifically the non-competitive inhibitor. So, yung imbis na dun sila pumunta sa active site ng enzyme, nahanap sila ng butas sa ating enzymes at dun sila napasok. Which is, pag nangyari yon, yung substrate na dapat na mag-interact sa ating enzyme, hindi makakapasok. And that's result why the enzymes will not function properly. Okay, so inhibitions caused by competitive and non-competitive inhibitors can be reversed. So inhibition by competitive inhibitors can be reversed by adding more substrate. So the higher the concentration of substrate, the more likely it will overcome the competition for the active site. So adding more substrate with non-competitive inhibitors has no effect on overcoming inhibitions. Okay, so reversing a non-competitive inhibitor requires a special chemical reagent to remove the inhibitor and restore catalytic activity. So here's an irreversible inhibitor, and it forms a covalent bond with an amino acid site chain in the enzyme's active site. While the irreversible inhibition, it also causes the substrate to be excluded from the active site. Okay. So, it is also a permanent inhibition. Okay, so irreversible inhibition is demonstrated in this figure. So, heavy metals, like for example, the silver, the mercury, the lead are examples of this type of inhibitors or inhibitors. Okay, so we have these antibiotics inhibit bacterial enzymes. So enzymes inhibitors are used to fight bacterial infections. Like for example, the penicillin. Penicillin is an example of an irreversible inhibitor. So it binds to the enzyme that bacteria use to synthesize cell walls and slows the growth of the cell walls. So without a cell wall, bacteria cannot survive and the infection stops. Okay, and that's it for enzymes. Hope you understand a lot of important facts regarding enzymes activity. See you all in our next lecture. Comment your name, section, and a short key takeaways. Thank you and God bless.